Yes, thank you. So first of all, I would like to thank you, Pierre, for inviting me to make this presentation this morning, saying hello to everybody who is here, and also to fill in for the organization in the name of NORA. My presentation will be dedicated to evolutionary history, convergence, and genetic parallelism in the European flat oyster. But I will introduce first why it is important in the context of restoration to have a better idea of the genetic structure, diversity and structure of this diversity in European populations. We know that it's important, this species is important for farming, far behind the cupped oysters, of course, but also in an ecological point of view for restoration of oyster reefs. We know that this species is a native oyster species, and it has been overexploited. It has been exploited for a long time and overexploited in the 20th century, beginning of this century especially. Sorry, I have to move that. Yes. And this species also faced several diseases, mainly due to parasites. So at present now, the range is far less as it could be observed here. You can see on this map the red area where the species is supposed to be present. In France, for example, now, the species is still present in several areas in France, but found only in Brittany. And there are several problems for this species because and it has been included since 2009 on the list of threatened and declining species and habitats by OSPAR. As you can see on this graph that comes from European Native Oyster Habitat Restoration Handbook that you can find on the NORA website, several factors are affecting native oyster life stages. I talked about overfishing, but pathogens, but there is also the invasive species like the cupped oysters, the environmental conditions, and so on. So clearly, it is now several groups and farmers are now dealing with restoration projects in France and Europe. And for the projects I know in France, there is one called Forever that is led by my colleague Stéphane Prouveau in Ifremer, Brest, and it's dedicated to areas in Brittany. And I know about another project called Refona that could begin this year and wants to see if there is possibilities of restoration projects in Marais-Oléron and Arcachon Bays. But you can see, or you will have presentations this morning, I think, about such projects, but you can find them all in the NORA website. I wanted to introduce the different questions that can be posed when there is an interest in restoration. And a recent paper gave a synthesis of that with several recommendations. Here you can see several of them. They encompass recommendation about the oysters themselves, about producing, of course, enough oysters for restoration or preserve their genetic diversity, but also about the sites, selecting good sites, I would say, free of bonamia, but also provide a suitable substrate for successful recruitment. What I want to emphasize here is that I would say that one of the prerequisites would be also to know, to have a better idea about the genetic characterization of the stocks or population of oyster we want to reintroduce. And in terms of genetic diversity, structure, maybe also resistant to Bernamiosis, and we'll have examples. So now I will give you recent results about projects we made about the genetic diversity at the European level. What we knew before is that we observed, I would say, a classical Mediterranean Atlantic differentiation, a weak one, but still significant. And we observed that with microsatellite markers or mitochondrial markers. And we decided to deepen this question by using more markers that we had developed a few years ago. And for the results I will present you now, we have been using 203 SNP markers. That means SNPs means differences between Oyster's genome of one base pair. So we have 200 of these differences. So we sampled oysters from, or we received samples from colleagues from Norway to North Sea, Atlantic area, Western Mediterranean area, Eastern Mediterranean, and Black Sea. The first result we had can be summarized in this picture with that is principal component analysis with the two first axes. And you will see that we have the code color here of the northern population in red, North Sea population, in dark blue, Atlantic population, light blue, West Mediterranean population, and orange in East Mediterranean or Black Sea. What we can see here is that a small part of the genetic variation is explained by those two axes, but it's still significant. And on the first axis, we can see a geographic effect, I would say, from the north to Atlantic, Mediterranean West, and Mediterranean East, sorry. And on the second axis, we see a second pattern with a grouping of those two clusters of population that are at the that are inside the range of distribution, and here clusters of population that are at the end of the range of the distribution. This is validated, or we observe the same kind of results with a structure analysis, still from Norway to Black Sea with the same color code. We still have 4 clusters. And here I just added this because you can see this Italian population that is dark blue, but we would expect it to be light blue, and it's here also dark blue. And our hypothesis is that this stocks population would have come from a stock coming from the Atlantic. And these could be corroborated by recent results obtained by colleagues in Adriatic Sea, where they observe that aquaculture may have an impact on the genetic diversity of wild oysters in this area. It's important to mention in the context of restoration, because we could have in that context the development of non-local stocks and maybe maladapted for different kind of traits. And we could also observe interaction between wild and aquaculture stocks. We wouldn't really know what they could lead to. So to go on the results of our study, I showed you the two first axis of the PCA, but when we look at the third axis, so here first and third axis of the PCA, we observe a strange kind of pattern. You can see 3 groups, or each dot is an oyster, and you can see this kind of strange pattern of grouping. And when we look at several markers, some of them like you have two examples here of two different SNPs, and we can see that it is a kind of grouping of their genotypes. For example, for this marker, you have the three genotypes grouped like this, and for another one grouped like that. So we observed a weak but significant structuration of the diversity correlated with geography, but certainly not only. And we wanted to deepen that. And this pattern made us think about the possible linkage of numerous markers that for which there wouldn't have high recombination. And we were thinking that maybe they could be in potential areas where they would be linked. So we decided to look at the linkage disequilibrium and coefficient of correlation between pairs of markers. So here you have the coefficient of correlation between all the different pairs of markers possible. You can see that most of them are not linked, but here you have sometimes high coefficient correlations for pairs of markers. Here we have another representation. You have the semi-matrix of all the different coefficient of correlations, and you have their value. So when it's dark, it means that there is a high coefficient of correlation. So it's small, but you can see that we have been able to detect several groups of markers that are linked, especially those two big groups. And those main groups of linked markers were confirmed in linkage group 1 and 4 of the genetic map we produced a few years ago in the context of another study. In this study, in fact, we produced a genetic map because we wanted to make the link between markers and resistance to bonamiosis. You will hear about that kind of work also this morning. But in this context, this linkage map allowed us to confirm that those markers are really at the same place, I would say, on the genome. So if we look at the allelic frequency of those markers, those linked markers in the different populations, so here again, you have the code color from red Norway to Black Sea. You can see that for this group, each time for each SNP, you have a high frequency in the North Sea and in the Black Sea and East Mediterranean. Sometimes a little bit less, but the pattern is clearly always the same. And it's the same for the second group of linked markers here again, especially with Norway here and the Eastern and Black Sea. So looking when we also look at the differentiation between population, especially between North Sea population, differentiation between North Sea population and Atlantic population on one side, and East Mediterranean and West Mediterranean on another side, we were looking for the markers that were explaining the main differences with FST parameter. We end up with all those markers, linked markers, that were outlier. It means that they were the main markers explaining those differences. So we decided to conduct a project called Flat Track in order to test for our hypothesis. It is what I mean by that is that I wanted to compare some of the populations that have revealed this surprising pattern of genetic parallelism, at the two ends of their range. And for that, we wanted to analyze entire genomes of few individuals of the different lineages we had, and using a long read sequencing technology. So for that, we need to get samples possibly fresh samples to be able to extract high molecular weight DNA, and this is not so easy. So Florence Cornet in my team is working on that. And after the sequencing, the idea is to be able to, sorry? No, the idea is to be able to partition those genomes into a multiple of chromosomal blocks from the different lineages. And then we want, so all this information, and then the idea is to combine all this information to be able to contrast the regions that follow the general pattern of geographic isolation and the regions that reveal this parallelism between distant populations. And so we want also to look for structural variation if they are present, because we think they could have a potential important role in this kind of evolutionary process. But as far as we don't still have all these samples, especially those samples available, we decided to conduct a first approach still at the genomic level, but using short read sequencing. So we decided to extract DNA from 28 samples from the 4 different groups we have, and also an outlier sample, and with NovaSeq technology. And the idea here is to try to reconstruct the evolutionary history of the European population. And for that, first of all, we try to confirm the pattern we have observed, the same structuration. We suppose we will get the same, and maybe more detailed because we have more markers. And we want to use the joint side frequency spectrum analysis with Paul Doniol-Valcroze, who is a Master 2 student who is working now in collaboration with ISEM, who develop the whole strategy, I would say. So I will give you information about how it works, very schematic way. So here you imagine that you walk along the genome, you stop on a window, and you look at the SNP. In one population, you look at the frequency of the SNPs, the different SNPs you look at, and you count the number of SNPs. Here, there is a high number of SNPs with a certain frequency, and you do that with all the different frequency from 0 to 1. You do the same for the second population, and you end up with a kind of thing, what is called this joint side frequency spectrum. So after that, here you have the number of SNP. You count the number of SNP with this different kind of frequency in the 2 populations. So here you will have, in fact, the private polymorphism of one population, the private polymorphism of the other population, the fixed difference in the corner, and in the middle, you have the shared polymorphism. So based on that, you will try to reconstruct the history based on this spectrum. So what about the history of those populations? You have several scenarios possible. And if you begin with an ancestral population, you can imagine what could have happened. You could have a divergence without gene flow. It means that the population is separating and they don't exchange any genes. You could also have that kind of thing, but after a moment of separation, you could have exchange. So it is what we call a secondary contact. And you could also have something like that, separation of the population, but with still gene flow. It's called divergence with gene flow. So I won't go in far details, but here just to show you that we could see different patterns. So for example, if we, for example, start with an ancestor population, shortly after the separation, the split. Most of the SNPs are shared at similar frequency between the two populations, but as divergence increases, more and more SNPs are in fact accumulating in the edge and the corners of the spectrum. This is what you can see here, here, and here. And at the end, you end up with a complete lineage sorting. So we can also, I could also say the same thing here, but I won't detail. But as you can see, we can have, when we look at those spectrum, different kind of patterns that we try to understand. At the genomic level, as I mentioned before, we know that there are regions that are not showing the same thing. For example, my two groups of linked markers, they show high differentiation between population, and we can make hypotheses about what is happening within each genome. So if we compare the genome of two populations, we can imagine that there are regions where gene flow is occurring and regions where they are not occurring. And in those two situations, you will have different kind of patterns. So this is what we are trying to do now, because we have now the sequences. We have been able to map all the SNPs on a reference genome, thanks to our colleague Arnaud Tanguy, that will explain his work. And after we know all those SNPs, and we are going to construct those spectrum. So I just end it.
Sorry, Sylvie,
yes.
Just to mention that it would be good to conclude if you want to keep some time for questions.
Yes, this is my last slide.
Great.
Just to remind you that we have been observing several clusters, 4 different clusters in Europe. But what would be interesting in the context of restoration is to know what is happening, I would say, in the contact zone between those 2 clusters, especially from France to Netherlands, because it is areas where projects are ongoing, and we don't really know how the different population are, the diversity is structured. It is the case on the East, but yes. Another question is what happens at the genomic level? It's what I showed you at the end of my presentation. And as short and long width analysis will come soon, I will be able to give you some results maybe in about 1 year. And I wanted to end up on the fact that I would like that we, in the context of NORA and restorations, that we would be able to promote that it is important to characterize the genetic status of the stocks available for restoration, not only for genetic diversity, but also for genetic structure, and also differentiation between different traits, such like resistance to bone amiosis. So thank you for your attention, and maybe it would have been a little bit too long.